So let's review some of the things we've learned so far about genetics. Phenotype is a physical trait, like height or skin color or hair color. Genotype is the combination of alleles, like big G or big G, let's say. Homozygous is when those two alleles are the same, big G, or perhaps little g. Heterozygous is when they're one of each. You have one of each type of allele. If I'm going to cross a big T with a little t, I'll just draw a Punnett square. Big T across the top, little t along the side. And then I'm just bringing the big T down and the little t over. Usually I use letters like T because it's easier to tell the difference between the two. So in this case, I've got a 50% chance that they're going to show the big T trait. So let's say that's tall. There's a 50% chance that it's going to be tall, and a 50% chance that it'll be short. Meiosis explains this law of segregation. Law of segregation, simply put, means that a parent can only give one allele to the offspring, not both. So here's our mother, let's say. So the mother, that egg cell is going to go through prophase, metaphase, anaphase, telophase. And we're going to wind up here. But because it's meiosis, we're going to go through prophase, metaphase, anaphase, telophase a second time. And now we wind up here with the haploid chromosomes. And you'll notice that there's only one allele on each one. That a parent can only give one of the two alleles. If this is the father over here, then in a similar way, the sperm cells only have one of the two alleles. That's the law of segregation. The law of independent assortment is different. The law of independent assortment simply means just because you have one trait does not mean you have another trait. In other words, the big R trait could just as easily pair with the little y as the big R trait could pair with the big Y. They're sorted independently. Just because you have one trait does not mean or guarantee that you're going to have the other trait. So just because you're tall doesn't mean you have curly hair. Here's a case of the law of segregation again, where the parent can only give either the little b or the big B, but not both. Independent assortment means that little b is just as likely to pair with a capital letter T as it is a lowercase t. And the capital B is just as likely to pair with the uppercase t as it is with the lowercase t. So independent assortment means the two are sorted independently of each other. Dominance just means that dominant trait shows up. Segregation just means that the parent can only give one of the two alleles. Independent assortment means whenever you're looking at a wide variety of traits, the one trait does not influence another trait. So let's look at some Punnett square problems for odd cases of genetics. Like, for example, multiple alleles. What if you have more than two options? So, for example, blood type, you could have type A, you could have type B, you could just as easily have type O. There's more than two options. You'll notice that the genotype, written this way, capital letter I with a capital A, is going to be the dominant trait. The lowercase I stands for O. That's the recessive trait. But you'll notice that I have more than one option. I have the A option, I have the B option, or I have the O option. So if I were doing a Punnett square and I was going to cross a person with type A blood, I could have a person that's type A that looks like this, or type A that looks like this. So if I'm going to cross a person with type A blood, let's say they're heterozygous, so AO. And they marry someone that has type B blood, and so we're going to put a B like that. Now again, I could write it capital letter I, A, lowercase I like that, but I prefer to just use the capital letters. So in this case, I'm going to put an AB right here, a B with the O right there, an A with the O right there, and then two O's. So there's a 25% chance that the offspring are going to be type AB blood. 25% chance that they're going to be B, 25% chance that they'll be A, and 25% chance that they'll be O. So here's what that looks like, the phenotype. So on the blood cells, there is a protein marker. That protein marker for type A, we'll say it looks like a triangle. For type B, we'll say that it looks like a circle. For type O, we'll say there is no protein marker there. In the case of something that's codominant, that means both alleles show up. So it's not one or the other, it's clearly both alleles are showing up. And blood type, again, is a great example of this, where we have both alleles showing up. In the case of type AB blood, you have both the A allele showing up and the B. So those phenotypes are both present on the surface of that blood cell. So type AB blood has both of those showing up. So let's say I were to cross something with codominant. So for example, roan cattle is codominant. So I have a big R, that's going to be red fur. WW, that's white fur. But if there's an R and a W, that's roan. Now again, sometimes people write this like capital H stands for hair, and I've got an R, capital H, capital R right there. The H stands for hair, but I usually just eliminate those and just stick with the exponents. So let's say I have a red-haired cow and I'm going to cross it with a roan bull. All right, so it's going to look something like this. In this case, I'm just pairing these R's here up, and then this R with this one over here. Then this R with the W below it, and this R with the W. And that means I'm going to have a 50% chance that I have a cow with red hair. But I'm also going to have a 50% chance that that offspring, that calf, will have roan hair, both red and white. Think of it like a candy cane. They're stripes, both red and white is present. Codominant. Let's say there's a case of incomplete dominance. Incomplete dominance. So that means we've got a blending of inheritance. Sickle cell anemia is this way. Like if I take straight hair and curly hair together, they might make an incomplete dominance. Wavy. It's neither straight nor curly. It's definitely a combination of both. And we can use flowers as an example of this. There's a lot of flowers that are incomplete dominance. So let's use the example of a flower that is purple. So I'm just going to use EE stands for purple. And I'm doing that because the letter P is really hard to tell if it's capital or lowercase. And we're going to say that H stands for white. And then if I have both purple and white, it's going to be a lavender color. So it's not purple, it's not white, it's lavender. Very light color. So let's say I were to take a white flower and I were to cross it with a lavender flower. In that case, make my little Punnett square. So I'm going to have lavender flowers 50%, and it looks like I'm going to have white. So 50% chance of white, 50% chance of lavender. What if I take two lavender flowers and I cross them? So in this case, I have a chance for purple, I have a chance for lavender, I have a chance for lavender, and I have a chance for white. So 25% chance for purple, 50% chance for lavender, and 25% chance for white. Polygenic inheritance just means more than one gene controls the trait. So all these interesting traits like eye color and skin color and hair color are polygenic. So there's more than one gene controlling it. So in the case of eye color and height, for example, the more dominant alleles you have, the darker your eyes or the taller you are. The more recessive, the lighter your eyes or the shorter you are. When we do that, we typically end up with a distribution that looks something like this. Individuals who are very tall, lots of dominant alleles. Individuals that are very short, lots of recessive alleles. The more dominant alleles, the taller you are. The more recessive alleles, the shorter you are. But generally speaking, most of us are somewhere in the middle. Some kind of blend of both dominant and recessive alleles. Sometimes traits are sex-linked. That means they're carried on the X chromosome. So, for example, hemophilia is this way. Color blindness is this way. So let's say that we have a female. She is normal. That's the H, but she's carrying hemophilia. It's a recessive trait, so that's the lowercase h. And the husband is normal. And then here's the Y. So normally what I do is I'll put both X's together and I'll put XY together. 50% chance for boys, 50% chance for girls. Then I'll put the trait there. So the chromosome from the mother is going to be H. The X from the father will be a capital letter H. With the other daughter, it's very similar. So here, that daughter could be a carrier. For the sons, this son is normal. This son would have hemophilia. So in the case of these offspring, the daughters, there's no chance that the daughter has hemophilia, but there is a 50% chance that one of the boys will have hemophilia, or a son will have hemophilia. That's sex-linked traits typically show up more frequently in men. So what needs to happen to have a girl that has hemophilia? So there's the XX for girl, XY for boy. For a girl to have hemophilia, she needs to have two lowercase h's here. So what does that look like in terms of her parents? What that means is one of those X chromosomes comes from the father, so he has to have hemophilia. The mother can be a carrier if she has hemophilia, which is a pretty rare case. Then any and all the offspring are going to have it. If the dad has hemophilia and the mom has hemophilia, all the children are going to have it. If the mom is just a carrier for hemophilia, well then there's a 50% chance that the children will have hemophilia. So that's sex-linked traits. They're linked to the X chromosome. Colorblindness is an example, and here's a quick little way to see about colorblindness. It's not that you see black or white. It's that you have a hard time distinguishing between colors. So see if you can see the numbers that are in those patterns. Sometimes the environment actually influences a trait. In other words, height and nutrition. You might have all of the genes to be a super tall person, but because you don't get a square meal, that genetic potential is never realized. But sometimes the environment impacts the genotype. So for example, in colder temperatures, typically turtles will hatch out as male more than they will female. So in this case, the environment is actually influencing the genotype in some unique species. So those are different ways that Mendel's rules don't apply to genetics. There's odd cases like polygenic inheritance, multiple alleles, codominance, incomplete dominance, sex-linked traits, and the environment can play a role. So I hope that helps you understand better some other non-Mendelian genetics.